somewhere around 75,000 years ago, don't worry, I'm not going to go too long on this, but around 75,000 years ago, human beings who were anatomically and behaviorally the same as us modern human beings, a few of them decided to trek on out of Africa and begin a 40,000-year project or so of populating virtually the entire world, becoming a global species, which we are now, perhaps the only global species, with I think the possible exception of cockroaches. I'm not positive of that, but I think that's correct. And now we find ourselves on the threshold of becoming possibly a universal species, or at least entertaining ideas of inhabiting places beyond this earth that we live on. So this new migration, however, I have to say, is different not only quantitatively from the original out of Africa diaspora, but qualitatively, because we're now talking about a migration to a place that has never influenced our evolution in any way. And so the question is, will we be able, like the first migration, to be able to do this by the use of technology and science without really changing our biology? Or will we now have to resort to changing our biology, to making ourselves in some way or another more adapted or more adaptable to environments that are even difficult to predict? So let me welcome our panel. To begin with, our first participant is a geneticist and associate professor at Weill Cornell Medical Center. Please welcome Christopher Mason. Also with us this evening, professor of genetics at Harvard Medical School and the director of the Consortium for Space Genetics. Please welcome Ting Wu. Good evening, Ting.
Hello.
Our final guest this evening is the director of astrobiology at Columbia University, where he studies, among other things, the conditions on other planets that make them habitable for life. Please welcome my friend and colleague Caleb Scharf. Oh, have a seat.
Thank you.
Good. I'm going to jump right into this with a question to Caleb, who said in his Scientific American blog that the possibility of humans living on other planets is, quote, terrifyingly hard to think about, and that's the best thing about it. What's hard and what's so good about it?
Well, I think some of these other people on stage are going to explain what's hard about it and what happens when you go to environments that are radically different than the one we've evolved out of. But what's good about conditions on another world or in space being so horrifically difficult? From my perspective, and what I was getting at when I wrote that article, was the idea that every time humans are challenged with something radically new, and especially in exploration, especially in survival, there's the opportunity for good things to happen. There's the opportunity for us as a species to expand not just physically across new territories in our solar system, but as a species, our conception of what it means to be human alters, and our conception of the preciousness of the origins that we have here on Earth, I think, expands and improves. So that was really what I was getting at, that this idea of, there are lots of motivations for our species to spread itself into the cosmos. I mean, there are motivations that are quite simple to do with potentially having a backup plan. Living on a planet is an inherently treacherous thing, especially on longer timescales than most of us experience in our lifetimes. So having a sort of off-world storage system and off-world backup plan is not a bad idea. And of course, the scientific enterprise, exploring the universe around us. Humans are pretty good at that. I mean, we build wonderful robots to explore the universe around us, and that gets us a long, long way. But there are many reasons to believe that putting a human in an alien environment is the single most efficient way of learning about new places. And I think the Apollo astronauts were an example of this. When you put a geologist on the moon, they did more in a couple of hours than you could do with a rover in years and years and years.
Chris, you think a lot about the future and long-term futures, which I find very intriguing, because I think a lot of our science tends to be, you think of scientific experiments that you hope you'll see the results of in your lifetime. So that puts a sort of a narrow vision. And one of the wonderful things about science for me is this kind of wider, bigger frontier. So you're making plans out for 500 years, is it? Is that what you're thinking about?
We're on year 5 of about a 500-year lab plan. I will
Good.
be
You're funded for that?
no, not funded for all of it. I will, of course, be dead for the vast majority of it. But I think it's actually really one of my favorite things. Well, first, it's terrifying for people who want to come in the lab. They think they're going to be there for 500 years. So that
I know graduate students just like that.
So not in the sense to have the experiment necessarily go that long, but actually to think about a multi-stage plan to learn as much as we can about the human genome and other sort of the ecosystem that we all are represented by, as well as ways to find sort of like I like to call do not disturb lists and places that we can alter to some degree in the human genome and the other genomes we have. Because I think we will have to have this list as sort of a parts list of the human genome, as some people call, or the areas that you can tweak, because we will probably need to do it for some of the places that we'll go. Mars is not a hospitable place, and so a lot of the plan is to learn as much as we can in a functional capacity in the near 20, 30 years, and then begin some of the engineering and long-term sort of tests of people on other planets. And I think it's a very quintessential human trait is that we all have an innate capacity to actually plan beyond our own lifetime, which as far as we know is unique to our species. Mm-hmm. And so I think it's your duty as a human being to at least have one plan that's beyond your lifetime. And the simplest example is just children. When people have children, is obviously beyond their own lifetime.
Students.
Yeah, students. Yeah. That's right.
So one of the places that I think actually makes a job of planning that is the Consortium for Space Genetics, which you direct. Ting. Could you tell us what you do at the Consortium for Space Genetics?
Yes, and perhaps the best way to do that is a little bit of history, how we came together.
Yes, please.
So most of us started in the Department of Genetics without a focus on space. But of course, many of us were inspired by space when we were young. So what happened is one day I went to a conference and I met Adam Steltzner, who is the person who put Curiosity on Mars. And we had a chance to talk a little bit. And he agreed to come and talk in our Department of Genetics because we knew that eventually the effort to go into space was going to intersect with the genetic information and genetic technologies. So my department actually was shocked that he would come and talk with us. And people were saying, why is he coming to talk to us? And then it dawned on everyone, because we are actually moving together to try and figure out a way to protect astronauts in space and to think about 500 or 5,000 years into the future. And what happened is that turned into one of the most exciting and successful symposiums we had in our department, with graduate students and postdocs leading that. And the faculty then, following the graduate students and postdocs, started thinking more creatively about the work that they were doing in their labs and how that work might apply to space. And that is how the consortium got together, sort of an excitement brought by a realization.
So can you give us an example or two of what of how this alters your thinking about genetics?
It became, well, genetics is moving very much into the realm of application and invention of technologies that didn't exist, and in fact, possibly genomes that did not exist. So much of the department focuses on disease, the detection of disease, and therefore information of personal genetics can help astronauts determine what their predispositions might be if they're going to go on a long trip in space. We also do a lot of work on understanding DNA damage and how to repair DNA damage. There's work on longevity, how to extend lifespan. And also, I work particularly on protection against radiation. And then, so those are all the things that are happening, and the goal in mind is to protect astronauts on space, but also take what we learn in space to take care of humans on Earth.
What about zero gravity? What about low gravity, microgravity? How is that? I mean, we know it has physiological effects. We could talk about those. We will talk about those when we get to the twins a little bit later, I guess. But is there any effective gravity on the genome, directly on the genome, on gene regulation, on gene expression, on any of those sorts of things? Either of you?
Yeah, I'd say, very much so. So as soon as you get into space, we see thousands of genes changing their expression. what we've seen in analyzing the data from the last couple of years of the study is that some of these genes immediately revert back to their normal state when you return back to Earth, but there are still hundreds that stay perturbed, that have lost whatever was regulating them, essentially back to the normal state. So we can see this is the short list of the risk genes, or the space genes that we're going to keep an eye on for the next, for every astronaut that goes up, probably.
Are these genes that we know what their role is, their function is, or are these just, this is just looking at, here are the genes on Earth, here are the genes there?
And the ones that change. A lot of them are,
some of them are
stress response genes, DNA damage repair genes. So there is definitely a physiological and genetic response to space travel, and that there is a stress response to a large degree. But one of the more surprising things we saw so far is that the telomeres actually got longer in space. And so we often think of our genome being
good,
fairly, the fountain of youth in space. You go up to space, and technically you're going closer to the speed of light, so you're a little bit younger than everyone on Earth. So you can be, and a little bit taller, so you can be younger, taller, lose weight, and be younger.
I'm going to sign up again.
Technically. But there's
going to be a tough night.
Increased radiation, your body puffs up, you have to eat, there's no shower, and you generally exchange all your microbes with everyone in a small aluminum can, basically. So those are the cons, I guess I'd say. But
I see. I'm glad there's no food here.
But there are some interesting genetic responses that we knew we'd see gene expression changes, because those change every time you eat, every time you sleep. There's a circadian rhythm, or even epigenetic changes, or how DNA is packaged. We know we expected that, but.
So I was going to ask you actually about circadian rhythms in space. It's a kind of an earthbound idea, isn't it?
Yeah. Well, every 90 minutes, essentially, you have a new sunrise. And there's really interesting things that we can also see in the data is when they go to sleep there's no up, there's no down, so they'll just kind of go in a closet and close the door just so that it's dark to get some semblance of circadian rhythm. But the other interesting thing is when you sleep, normally there's enough currents in the air, because we're all terrestrial beings, that you get enough oxygen flowing by you and the air moves. But if in space, actually, you'll end up sleeping in a cloud of carbon dioxide. So actually, we can see even there when they wake up in the morning, essentially they've been sleeping in their own basically gas exchange excrement, and then you can see it in the physiological responses that they're slightly suffocating during the night. So there's actually a race to see who can get the best fan action in the space station to avoid that.
Well, it's that hard to put fans in the space station?
Well, they're loud. Some of it's the noise, but you get used to it after a while, I think.
Caleb, you're a physicist. Is there a solution to, maybe we should make more gravity? Are there ways of producing at least minimal gravity environments?
In a space station, spacecraft, you can always do things like spin it up. So this is something that, if anyone's ever seen 2001, right? You remember famous scenes where he's jogging around this great ring that encircles the spacecraft, and the ring is spinning, and that centripetal force can be used to mimic the acceleration that you feel here on the Earth, or some fraction of it. And I guess I'm not sure it's ever been actually deployed in space. It gets kind of tricky, though, because unless you build a really enormous spinning spacecraft, hundreds of meters across, you can certainly spin something smaller, but you have things like the Coriolis effect to worry about. So if you reach across to grab something, where your hand is now in a different moving frame than where the rest of you is. Or you throw something to your colleague across the room, it'll kind of go woo around a corner. So it introduces other complexities to living in space. But my understanding is, and I don't know if this is really true, because it's not a perfect mimicry of gravity, that you can certainly create a form of artificial gravity by spinning up the environment and living on the inside of a spinning ring, and that may help mitigate some of these physiological changes.
So the space station, and that's sort of one thing where there's really virtually, I guess, zero gravity. But if you were to be on Mars, you're not in zero gravity anymore. There's less gravity than here, but there's gravity. Does it make a difference, or it's still a problem? Are we that finely tuned to a gravitational pull that even a small difference makes a difference?
I think, I don't know if we know, actually. I mean,
Yeah,
the moon is
right, I guess.
1/6 the gravity, and
Right,
people that were there were only
There
there
for a couple days, right?
not that long. And so, but the body's quite adaptive. You can see, I mean, on the way we solve it now on the space station is just with bungee cords. And there's a guy who I met at the Human Research Program meeting, and he's, you meet people, and he says, well, I'm the guy who designed the treadmill on the space station. I was like, oh, that's great. It's really cool to meet that guy who did that. And, it's obviously, it's a really cool device, but with a limited market in terms of sales. But it was an amazing device, and it's a bunch of bungee cords, a treadmill that's really rigorously designed, but that is the closest they can get to simulating gravity. And in the metabolomic data, or what's essentially present in the bloodstream, you can see both the markers for bone density loss, actually the physical loss, and calcium shows up in the urine, as well as all the enzymes activating for bone deposition, which essentially are really activated every time you have something like gravity. And so it's an ongoing battle on the space station for the astronauts today just to maintain what they have, but it's hard. And I think if you're on Mars, I think the first generation of people there would adapt to some degree. I think the big interesting question would be what happens in the F1, the other second, third, fourth generations.
Right.
And that, there's been a lot of science fiction written about how the people would be taller and misshapen and look like me in the morning kind of thing. It's, what they look like.
So there are several ways. So I guess we could get into that a little bit. And I was going to save that for later, but since we're here, we might talk about that. So that would be sort of an epigenetic effect, right? That by a second or third generation, you'd begin to see alterations in at least gene expression, if not mutations in genes themselves, as a result of some of these environmental effects. Is that kind of the idea that we're talking about there?
So, right, we're actually moving into the realm of Lamarck, where
Yes.
where a lineage will pick up a
Knew
trait.
he'd come back eventually.
Actually, I think Lamarck is coming back because this is a proposal that an individual organism, because of environmental stresses or experiences, will experience some stress, and then its progeny will actually inherit the response, let's say, that the parent had. And that was looked down upon for a very long time. However, I can tell you, I was at a meeting recently, and finally someone yelled up from the audience and said, just say the L word. And the speaker said, okay, I'm talking about Lamarck.
Yeah.
And there is evidence now from many areas to say that an individual organism may be able to pick up a response or a trait that will appear in the progeny in an altered form or a slightly modified form. And
then will be,
it will be passed
heritable,
on.
right?
Right.
Yes.
In some cases, it's heritable for 3 to 4 or 5 generations, and then the absence of the stimulus will disappear. In other cases, I guess people think that it can be more permanent.
And even in worms, we've seen that the multiple generations, if you have viruses that will infect some of the worm C. elegans, there's a model organism, a small nematode. For about 2 to 3 generations, they still maintain some of the defensive microRNAs, but then they'll slowly fade away. So even we think of DNA being, epigenetic changes being heritable, but there's even some evidence that a memory of the RNA response to a viral infection is heritable. As a paper from Oded Richavi 3 years ago that showed this. And so it's interesting to think that there definitely is a cellular memory that is present, what often we just call imprinting, but it seems to be more complex and more pervasive than originally thought.
I have a question.
Yes.
Am I allowed just a
Sure,
random
why not?
question
All right,
since these two
question,
guys
yes,
are sitting?
all right.
So thinking about, let's say, going to Mars, people going to Mars and living on Mars, and one of the things that interests me, I know nothing about it, but it seems like an important factor, apart from their internal microbiome, is the environmental microbiome. And let's assume that Mars is abiotic at the moment, that it's truly sterile. Then I guess there's 2 questions. Should we be bringing along an environmental microbiome to live in? But if we bring it from the Earth and we dump it on Mars, which has less gravity, a different radiation environment, is it going to go through its own epigenetic change and not be the thing we want to be surrounded by? I mean, I know it's very hard to predict, but...
Well, there's some evidence. We know bacteria in space become more virulent and have more resistance to antibiotics. That's been published by multiple groups. There's even an analog even in hospitals. If you look at where the most antimicrobial resistance markers or the antibiotic resistance, it's actually present more on the surfaces than it is of the patients from a paper just published 2 weeks ago. Writ large, the idea that they're under such strong selection pressure, you might be enriching for the organisms that might be the most resistant effectively on these surfaces. And so it's been one study, but it had 1,500 samples, so it's really an intriguing thought. So to your point of when we send people anywhere, that you're sending an ecosystem, not just a person, as well as what their environment. And they'll be under a lot of more intense and faster selection pressure as soon as they get to Mars. So I think we will have to have rapid diagnostics. You can't send a sample back to Earth to see what the bacteria is. You'll have to sequence it there on Mars. And one of the things we worked on with a team at NASA was just to do the first DNA sequencing in space. And so it's now possible to actually do diagnostics of what it is. So if you had to pick from one of 10 antibiotics, you might be able to know right away which one to pick instead of guessing.
Chris, I heard that NASA has a very, very stringent sterilization process for all the equipment that actually is one of the difficulties for designing equipment. And that also, many years ago, a group of individuals or nations got together to designate certain planets or certain, I guess they banned the contamination of planets. What is, and I think that's being reassessed. What's the status
The
there?
Planetary Protection, basically, it's an organization within NASA as well as an agreement between ESA and JAX, and NASA is too. As the name sounds, you want to protect other planets from us, vermin and filth, that will go out and distribute our filth. But really, they are obviously for discovery purposes. They're relatively pristine environments, and you don't want to have discovered alien life, but just have discovered what you brought with. I actually just had a long email exchange yesterday with a guy who sequenced an organism that was cultured from the space station. He thinks it's alien because it had evolved to be different enough from what's on Earth, and we're debating about whether that's true. It's a strain of bacillus, and they're funky to begin with. But the idea is that I think it's interesting for two reasons. One is that it's probably already failed. If you sequence the DNA in clean rooms, these are the samples we're getting in 2 weeks from now, there's DNA everywhere. It's literally impossible to get rid of the most abundant biomolecule on this planet. The DNA is everywhere. If you look at where it's coming from, and maybe cellulose is the most abundant, but in terms of a information content molecule, it's everywhere. It's in the air. It's in the atmosphere. It's deep underground. It's in hydrothermal vents. There are bacteria. They've had billions of years of a head start, so they're everywhere. And we've seen, if you take the blank samples from the clean room, they're not actually always clean. So on the one hand, we think maybe we've already failed or succeeded, depending on your perspective. But the other thing is, I think eventually, if you think long term, this idea of planetary protection is fallacious from its foundation. You actually, if you think long enough ahead, let's say 3-4 billion years, eventually the sun will engulf the inner planets. And so let's say we've done a perfect job and we have wonderful world peace on Earth and it's kumbaya every night and there's no famine, there's no disease, there's no war. It's wonderful. But if we're all still here on Earth, we will all die if we're still here in 3 billion years. So again, it's a ways off. But it doesn't matter if you've protected all the planets. They will all be engulfed by the sun. So if you really want to think about long-term preservation of our species, as well as all those of which we're responsible for, at some point you'll have to go to another planet and therefore lay down the microbes that were never from there. So the autochthonous state we live in here on this planet will have to eventually go to others. Or if we don't, it'll all go away, except for what we've broadcast into space. So I think it's a nice idea that eventually we'll have to fail. Right? That's how I would describe it.
Now, but so far, most of what we've talked about is sort of haphazard in the evolutionary sense of haphazard. That is, these things will go out there, they'll survive or they won't survive, they'll undergo natural selection, maybe some sort of epigenetic modification if they're lucky to get through a couple of generations. But what about the possibility of actually designing things to go out there, which we can do now even to ourselves with techniques like CRISPR-Cas9 and gene drive? Maybe you could explain a little bit of that for us and whether or not this is the kind of thing we should be thinking about and doing.
I definitely think this is something we should think about, not just for space application, but
Of course.
on the planet. So people have been modifying genomes for a long time. We have quite a wide variety of ways of doing it. But there are a couple of things that made it not reasonable to think about modifying in any practical way. And one is the targeting, and the other is the efficiency. Those were the two things that
By
held.
targeting, you mean?
Targeting, you want to modify this part of a chromosome. You want to just modify that, and you want to do it efficiently. And until recently, we did not have the machinery, the enzymatic machinery to do that. And then just a few years ago, 2013, through this technique, this molecule or this mechanism called CRISPR, we now have the efficiency and much, much better targeting, as well as two other very good technologies for modifying genomes, zinc fingers and talons. So this triumvirate of machineries really give us tremendous potential to modify genomes. And I think probably many of you have been involved or read articles or been involved in conversations having to do with just how much do we want to apply this. I would say it's really almost as if our species has moved into a new era. We passed a milestone where we can now intellectually understand ourselves before even an individual is born, and have a better understanding, not perfect understanding. And now we have the capacity to design. So,
And the responsibility that comes with it, presumably.
and the responsibility to, right. And it brings up the issue of judgment. We now can think about genomes and judge them based on all different kinds of measuring sticks. And this is what I think the world is thinking very hard about. We need to involve all different communities in this.
I'd like to talk about the twins experiment now in space. So let's back into space a little bit, which you're very involved in. So maybe you could tell us a little bit about Mark and
Scott Kelly.
Mark and Scott Kelly.
So for those not familiar with the study, it was a plan to, it was really a unique opportunity because we had twin brothers, both of whom are actually astronauts. And so could you imagine being, I don't know if any in the room are parents, but like, could you imagine being a parent, like, oh, both my sons are astronauts? People be at a party, be like, yeah, whatever, it's not really true. Like, no, really, both my sons, my twin sons are both astronauts.
So there must be a gene for it, right? Because
So
the twins have it. I mean, clearly, there's an astronaut gene.
We've confirmed that they're twins. We did the genome sequencing. I can tell you that for sure. That's been definitively confirmed. The plan was, it was actually launched at a press conference because Scott Kelly was going up for the longest American mission in space, which is a full year. And when they were planning the mission, oh yeah, you can see them up there. As they were planning out the mission, they said, you know, actually it was one of the reporters who said, well, you have a twin on Earth, are you guys doing anything with the twin? And they said, well, we don't have anything immediately planned, but as soon as the press conference finished, so here's a big high five to the press to ask good questions, is they said, well, maybe we should do something. So actually the twins themselves really proposed it to NASA and said we should do something. So they had an announcement to have people submit applications. And I had actually sent an application to do astronaut genomes back in 2010 when I had just started my laboratory. I actually wrote this really interesting grant. I said, I don't want any money from you. I just want DNA. And they hadn't yet been banking DNA from astronauts. Because normally when you start a lab, they give you some funds to get started. So it was a peculiar grant. But they said, well, we don't have anything banked yet. But then eventually, when I knew they made the announcement, I said, oh, great, I've got most of it written, and submitted and was one of the labs selected for. They said, okay, well, we have identical twins, and we're going to monitor them everything we possibly can, because the big question was, well, what changes in the body when you're in space? And so we said, well, no one's ever looked, so we don't know. So we should look at everything we possibly can to understand the physiological changes to the body. So we're looking at the DNA, what changes it genetically. Epigenetically, we talked about the modifications of DNA, what happens to your RNA molecules or your transcriptome, as well as the modifications to RNA, which we talked about, protein changes, metabolites, how DNA is wrapped and packaged in all your cells, microbes on the stomach and the skin and the mouth, and also in the environment, looking also at what are called metabolomics, or small molecules in your blood, cognition, how fast actually they can do tasks. We're also looking at vasculature and doing full body MRIs, and doing all these tests, and we did over 19 time points. So the amazing thing about the study was not only, it felt a little bit like a Michael Crichton novel, if you remember like some of Michael Crichton's books, where they get a peculiar mix of scientists, like we need a mathematician, and we need a geneticist, and we need a physicist to go to Jurassic Park. You know, it's like a weird mixture of people. And there too, we have a telomere biologist, some geneticists, some proteomics experts, a guy who spends all day designing, you know, computer games for cognition testing, cardiologists. You know, so it's really, it's a team effort, and you know, I'm just one of the team. But it's also forced us to think about systems biology. So if we want to measure all of these changes and then integrate them, there actually is not really an infrastructure that exists either for medical records at hospitals or for astronaut monitoring to integrate all that information. So it's been a computational, exciting challenge. You talked about challenges before. It's great because it's the most challenging integration problem that we have. But then also some of the biology has been really interesting. We've actually seen already that your telomeres get longer in space. The stress response genes really activate. The microbiome inside your gut changes, but that wasn't too surprising. They're also eating different food. And we can also see, some of the responses to the hypoxia, the changes in whether you have changes in oxygen and carbon
What
dioxide.
was the Earthbound brother on the same diet and all the rest of that?
So
Or
we tried to get Mark to have the same diet, and he politely refused, or actually not politely, he definitively refused, and said, "You can go." You can imagine the rest of that sentence. But we did get them to both keep food logs, and his log though was more, So Scott's up in space. It's highly regulated food. It's rehydrated and then heated. There's a little bit of an exchange with the Russian side, so they do exchange some food and other odd things we keep track of. So the Russian water supply is different from the American and European. The Russians put a little bit of copper into their water supply, whereas the Americans don't, but they both try and get rid of microbial contaminants. So we have to track that as well. But Mark's food log was more, burritos and margaritas, and it was a very terrestrial
wait,
diet.
they don't send them with any booze up in space at all?
You can get a care package.
Oh, now
but
I'm
you can
definitely
get
out. But are there any benefits to being in space? Does anything good come of it for your genome, your biome, or anything?
This is an ongoing debate amongst all the twins investigators, because we keep getting this question, because all we keep talking about is permanent changes to your microbiome, epigenetic changes. The only potentially benefit might be is the telomere lengthening, and actually they come back in pretty good shape. It's because they have to exercise every single morning for at least 30 to 45 minutes just to maintain their bone density, and so it really
Did
is.
you get the Earthbound brother to do that?
No, again, no. The margarita.
There are a lot of
margarita.
controls
Yes.
missing in this experiment.
So we do often joke incorrectly that this is the worst powered study in human history. It is an N of 2, really an N of 1, because they're twins. And so this study was never designed and never could be the definitive measure of what happens to the body in space, writ large. But it is the first time NASA has gotten into genomics. It's the first time they've really wanted to think about precision medicine for astronauts, which they have a nice poster now that models all this. And they want to begin to look at the question of, well, if we have a genome, could we think about who's at higher risk for hypoxia-related genes, or those people that might respond better or worse to different pharmacological treatments in space? So they are doing very much what our healthcare system is, having a genome-guided view of medicine, your genome and your microbiome.
So would that ever reflect back on who could be an astronaut and who couldn't, do you think?
This is an ongoing point of contention within NASA. So the IRB that we got for the twin study itself took
It's an institutional
months.
review board document you need for human experiments.
Yeah, before you do any experiments on humans, you need to have a board to go through and make sure you're not doing evil things to them, as you can imagine. And there's unfortunate history in genetics and in eugenics where really bad things were done without institutional review boards to make sure you're informing the patients of their risks and making sure that all the experiments are medically necessary and that you have good controls. So this was the first time it's been done for NASA, and we want to use this for subsequent astronauts, but they don't. The flight surgeons and the astronauts have a kind of a love-hate relationship, more kind of hate-hate, is that the flight surgeons are always looking for something to potentially, maybe that's a high risk, but if they flag something, it has to be enough of a risk that they'll keep someone from going on their mission. And all the astronauts have trained for years, some cases decades, and it's their life's dream to go to space. And so they will do things that you would imagine humans would do, is the dosimeter, which tells you how much radiation you've had, they'll hide it in the space station so they can go back up for different missions. They'll keep it in a protected part of the space station, or they want to go back up. I mean, it's their dream, right? So if you want to,
Is it addictive? I mean, is there a space addiction?
We could
came
look
up
for
with that
addiction
yet?
genes.
Yes,
There's no
definitely.
addiction. Look, there's a big debate in NASA of how much can we really reliably tell someone about whether they should or should not go to space just based on their genome. And most of the consensus is we're not there yet that we could say. But it's a question of, I think, when, not if. And it's like a little bit like the movie Gattaca, which most people in this room have probably seen. But it hopefully not the bad version of that. We're hoping it would be all the information that you could do to protect yourself. And for example, from your genome, you can tell what drugs you should or should not take that you would respond well to or not, or your microbial genome. So hopefully we would use the good of genomes to reinforce good genetic circuits as well as even protect them, is the hope.
I think the situation is made more, put in starker relief because the trips that people are anticipating now are longer. And so if you're going to be taking a 10-year journey, then you can actually, for some cancers, calculate a predisposition or a chance that you are going to develop a cancer in a very long period of time. And you'll be too far away to come back for treatment. And I think that's where the discussion of genetics, personal genetics, has become a little more interesting.
And that there, you have to take it into consideration. It would be foolish not to. So it's a big if all the evidence is solid and grounded and replicated and really informative, then it should be used. But it's getting all the scientists and the flight surgeons at the point where they all feel comfortable making that decision. But I think when you think of missions to Mars or longer missions, you can't get back fast.
So I thought go on.
Oh, no, well, I mean, this is very interesting. And I was thinking about Mars specifically because, really the only way to learn the peculiarities of the Martian environment and its impact on humans is going to be to send that first batch of, ideally many, perhaps all twins, to do that experiment, because it will be a very different experience than low Earth orbit. The radiation environment en route for, let's say, 6 months will be different, and the stresses of that will be different, and then the experience on the surface, which no human has yet set foot on Mars, so we don't have the slightest idea. I mean, it's a different chemical environment that you're going to have a hard time keeping out of your potato chips if you stray outside of the habitat and as in the Martian. So that's interesting. I mean, it sounds like you would have to have a biological expedition would be one of the first things to do.
Well, I have a question for you. Do you think that it would be a good intermediate step to not to focus, of course, to try and get on the planet and live on a separate planet, but to actually create a space station where we would be able to control the environment better, rather than focus entirely on trying to get on a planet or alter ourselves to live on that planet? Your knowledge of the universe, what do you think is?
Well, it's an interesting question. I mean, I suspect from the practical point of view, from a sort of rocket science point of view, it may be equally challenging to construct let's say, a more elaborate space station that, say, mimics Martian gravity, for example, because that's something you would want to do for it to be worthwhile. Also, if it's in low Earth orbit, you will not be exposed to the same radiation environment. So you're really talking about, it sounds like a reasonable idea to me, but it would be a structure, a space station that would have to be, maybe it would be orbiting the moon or something like that, to alter, to get a little bit closer to what you need for that experiment.
Well, actually, I was thinking more that rather than trying to find a planet where we could thrive in make it like our environment, just to perhaps forego that until we find a planet that is perfect for us, and instead work on building a station that is just like Earth. I
mean, unfortunately, if there are planets perfect for us, they're a long way away around other stars. So then we're into the whole realm of interstellar exploration. And, this is something and this circles back to my first comments about, we have this picture, Hollywood is partly to blame, and also our wishful thinking over the years of, going to Mars. Mars is the other habitable planet, and of course it's an appalling place. There's almost no atmosphere, it has less gravity, it's got nasty surface chemistry, it has less shielding against ultraviolet radiation and cosmic radiation. It makes me think back to, this is a sort of little anecdote about how we get so fixated on certain ideas. Going back to the very first robotic spacecraft to go past Mars in, I think, the late 1960s. Mariner 4, I think, was the first flyby, and then there was Mariner 7 or 9 was an orbiter. The first US orbiter to get to Mars. You can actually go and look at the original scientific literature, and people like Carl Sagan, who was active at that point, was literally writing papers speculating that, well, when we get to Mars and start taking the high-resolution images, we may see this pattern of vegetation. If there's vegetation, there might be certain types of herbivores and so on. And of course, well, two things happen. First, they get to Mars, and Mars is encased in one of its global dust storms. And so all that could be seen, the first close-up, high-res orbital images of Mars were the peaks of Olympus Mons and a couple of the other Tharsis volcanoes, and the rest of the planet was covered in dust. So they actually had to wait a couple of months. I mean, it must have been really, I mean, it's kind of a science fiction-y thing. You know, you're waiting for the clouds to clear or the dust storms to clear to reveal more dust. And a terrible
Where
planet.
do they think all that dust came from? I mean,
Well, yeah, you know. So I mean, I'm sure
When the dust
people.
settles.
I'm sure people speculated. So that's just by way of saying that. Yeah. We, you know, in our own solar system, there aren't that many planets. So, coming back to what you said, I agree. I mean, it's so the engineering challenges are different. There's some advantages of building a raw space habitat for yourself is you don't have to lift it in and out of a gravity field all the time. Getting stuff down onto Mars is a complete nightmare because you don't have much of an atmosphere to break yourself, actually. And this is why the last big rover couldn't use airbags to bounce on the surface and do the final slowdown. It had to use some kind of rocket crane system because there isn't enough atmosphere. There's enough gravity to get you to the surface hard, but there isn't enough atmosphere to use as a natural brake. But, you know, what's interesting to me, the idea of a fully contained space station is, you know, do we know how to do that for the long term? I mean, the ISS, it gets resupplied. You know, you can flush out things every so often, I assume. You can.
Sometimes when you see a shooting star at night, it's not a shooting star. They
I'm not
eject
going to
away.
ask what it is, but I can guess. Can you tell if it's Russian or American?
Different emission spectra, you can see the colors. Yeah. It's all vodka. Yeah.
So I think it's a fascinating
that took
question.
the romance out.
Yeah. No. But I think it is a very, very interesting question. But it is an immense challenge, and if you actually add up the total mass that humans have elevated from the surface of the Earth and put into space, it's not very much. I mean, I did this calculation at some point, and I think it's some tiny fraction of the mass of an oil tanker has actually been lofted into space, even though there are thousands of pieces of satellite and junk floating around. It's really expensive.
And not that many people have actually, about 467 people, or actually not 472, have left Earth and then come back. There's not that many people even that have really experienced it. But it's a good question. Maybe the self-contained environment or waiting until we find the right place. But even if it is the right place, you'd have to send people there. A lot of them would probably not make it. And I think our intuitions about this, where everyone wants to go there and have it be, kumbaya and be fine. But you think of like the people who first came to Plymouth Rock in the first winter, more than 90% of them died that winter, but more came the next year. And so I think the entrepreneurial or pioneering kind of spirit may require, or historically has required, a lot of sacrifice. And there's a contrast. When they came to Plymouth Rock, there was an atmosphere here, there was water. It was a little easier, but they're very. I mean, I think NASA has a lot of really great homages to the astronauts they've lost over the years, and but they all recognize it's a really high-risk job, and I think it'll be true whenever and however we send people.
I mean, another possibility, and this is something I think I've even written briefly about. There are places on Earth you could begin to test some of the, and it's not just the desert somewhere, but set up your colony on Everest. At the top of Everest, right, where people usually die, right, because there isn't enough oxygen to sustain you for any great length of time. There's a lot more UV radiation. There are no natural resources. You can't grow tomatoes, right? I mean, so there are some perhaps interesting experiments that could be done here on Earth, not just building a pretend Mars habitat in Iceland or something, which is interesting for testing the psychological aspects of isolation and so on. But we could do a little bit better here on Earth.
was talking with a physician at NASA about the immediate problems: bone loss, muscle loss, kidney stones, visual problems, blood pressure problems, and but for many
Sounds
of
like
these,
me.
You're not even an astronaut yet.
I'm doing that all on my own.
what the physician said, well, see, so it might be, you might not, this might be more encouraging for you.
I'm waiting.
They said these problems are very complicated for the first few days or first few weeks. But then, interestingly, the human body acclimates. And they say even bone loss and muscle loss is appropriate for space, where things weigh less, and where, in fact, they say you get calluses on the top of your feet because you're using the top of your feet to hold you in place. You lose the calluses on the bottom. And they say after a while, the human body acclimates for long term. And the problem is when they come back to Earth. So these habitats that we build and these things that we think are terrible for our body, in the long run, he was saying, actually, the body may be adjusting just the way it needs to adjust for long term.
Even Scott, when he came back to Earth, he one of the hardest things that happened to him his first day back was that the weight of clothes made his skin hurt. And so he, because he hadn't felt the weight of just clothes, so he notoriously spent some of the time just wandering around the house, native, and just to it was painful to wear clothes, which.
How does his brother feel about it?
They weren't Skyping or FaceTiming that much, I don't think.
But he was fine up in space.
Yeah,
before he
Yeah.
came
So
down.
the trick is to send people out there and not let them come back. Then, in other words, right? That's it's just a one-way ticket. Okay. With that, let's take some questions. This is a salon, so we'd like to hear from you guys as well. If that's okay. Good. So we have some questions. Yes, give him a try before he before he blasts off.
So, hi, I'm Ishan, and my question is, so you talked in the beginning a lot about like biology and how human genes could make humans adapt, but like if someone went to Mars, right, and they were like having a baby, then the baby would develop differently in the fetus. They would have maybe weaker bones and less muscle mass and stuff like that. And what would do you think they'd be able to return back to Earth with the weaker bone mass and the weaker muscle mass and the like never being experienced to the high gravity and high atmospheric pressure of Earth?
It's a great question. The answer is it would definitely hurt when they came back here. I think it wouldn't necessarily be impossible unless it was thousands and thousands of years. It could eventually become literally a different species, quite possibly. Where if they were to come here, it would be too painful, or they would then need spacesuits to come to Earth, and they would be just have been our cousins. So you could very well imagine that with enough time, they will become a different species. So I think that it's actually inevitable. I mean, even if just people living in the South versus in the Northeast have different accents, and that didn't take that long, really. So it'll start with language, it'll start with culture, it'll start with intonations, and very quickly it manifests in the biology. And the selection pressure will be so fast and so strong. I think it'll be an extraordinary evolutionary experiment on our own species. But I think there'll be changes.
Although it sort of raises a certain irony. These ideas, for example, Elon Musk talking about colonizing Mars as the backup plan, except they won't be human after a while. So it's not quite the backup plan you had in mind.
And he wants to get there before NASA wants to be there by 2033 or 2035, and he wants to beat them. And so there's a bit of a battle between them. But it should be soon. Actually, you would be about the right age to actually be one of the first people to go to Mars. So hopefully that's
You can sign up in the back of the room.
Some one-way rockets. One-way rockets.
We have another.
It's so hard to visualize the future. It's so far away, and there are tons of problems that come up immediately. And I'm kind of concerned that there's a lack of discussion about it because obviously everybody has different experiences and views on ethics and stuff. And I was wondering what kind of people you think should be in charge of dictating the ethics of genetics and balancing future thinking with present-day problems.
That is such a timely, perfect question. I think that geneticists realize we have, as I mentioned before, moved into a new era. We have the capacity to really change the way genetics enters our lives and how it affects our lives. I think we all recognize also that we are in service to the world. It's not, it's less and less what we think is fun to do. When I was a student, it was how to figure out how things worked. How did this happen? How did this evolve? Very much now, it's turned into, we know enough of how this works that we have the capacity to invent something. We know enough about the basis of this disease and the location of this disease that we can think about changing it. And so we're no longer solidly in the discovery mode. We're in the mode of altering our environment. You are our environment. We're all part of the environment. So I think you will find greater agreement among scientists that this is a world problem and is going to have to be solved by world discussion. Are there certain people that will be chosen? I certainly hope not. I think, and the more noise people make, the more opinions you voice, the less chance it's going to be made by a small group of people. I don't think we want to make it with a small group of people. It rests really on how much involvement all of you take onto yourselves.
I think I'd echo it. Definitely is so far, I think, and should be a community effort. Like the scientists talking with everyone in the community, religious scholars to people working in every different discipline. And one interesting thing is I think it should not be just left up to the scientists themselves. Actually, at the meeting we were recently at, the Genome Project Right meeting, there was a survey during the meeting of one of the questions was, do you think scientists are doing enough to mitigate potential risks and think about the ethics of the problem of engineering genomes? And 70% of the people at the meeting said, well, yeah, I think so. And I said, well, that right there, I've tweeted that and said, well, this is a reason why we shouldn't just depend on the scientists, because it's too easy to fall in love with the data and to see the awesome power and potential of the biology and the tweaking of it to improve life and eradicate disease. You can easily get caught up and think, well, this is great, and yes, we're thinking about it and we have panels, but I think it's not going to be enough. And most of the people at the meeting, while optimistic, very much welcome feedback, comments. They want everyone to be involved. And if they don't, then they're working with Dr. Evil.
I would say I welcome the idea in many ways that that we'll once again begin listening to philosophers and historians in science and not just technicians, as it were. So I think that's actually I see this optimistically, I would say, if we get that back in the mix. Questions, more questions?
If we're as a species going to continue, we are going to be thinking about millions, even billions of years into the future. So, and there will become a point where even Mars is not a habitable place for us. So expanding on to outer reaches of the solar system and beyond, and that the farther we get apart, species of humans will become isolated and possibly speciate. But
are you thinking about the heat death of the universe, and it keeps expanding? I think if you keep going, eventually that's where you'll end up, for sure.
But more the idea in that we will, at some point, hopefully, be expanding outside of our solar system, beyond the influences of the dangers of radiation from our own sun. And my question was, what influences are humans going to have in deep space on like our physiology and living, beyond the influence of our own sun?
The radiation environment of our sun is certainly an issue, but there's plenty of so-called cosmic radiation that comes from other sources throughout our galaxy. It comes from other stars, it comes from black holes consuming matter, it comes from neutron stars, it comes from particles accelerated in galactic magnetic fields. We have a bit of an idea of what that environment is like. It's pretty similar to sort of deep interplanetary environment in our own solar system, so it's kind of nasty.
Not any safer.
It's probably not any safer, and yeah, and also just to add to that, I mean, if we're just thinking about it with an element of realism, to get to another star in a reasonable amount of time. Now, we may have to engineer humans to live longer to do this efficiently, but you will have to move fast. And moving fast through interstellar space brings all sorts of hazards that are quite mechanical. I mean, you may spend most of your time patching up your colony ship because a microscopic piece of interstellar dust has zipped through and, went through Stu's cocktail, and but I think it's the radiation environment I think is pretty similar to deep interplanetary space.
So I know geneticists are trying to identify the genes involved in DNA repair, but we also are looking for ways in which the genome, which plays a big part in our life, can withstand onslaught. My lab in particular is interested in some very mysterious sequences in all of us. People don't know what they do. They're the least evolving sequences known by definition. They've been in all of us for 300 to 500 million years without change, essentially without change, and they're a big surprise for genomicists.
The do not disturb.
Right, do not disturb.
Yeah.
But recently, some of our work has shown that, I suggested, that they are like, we call them genomic canaries. They're scattered all over your genome. And what we think they do, it's a model, and when they detect a damaging break in the chromosome, they get rid of the cell that has that break. So I think it's possible, as we explore our genome more and more, we'll find the tools to be able to fashion ourselves to withstand radiation damage. Perhaps we're also studying in the lab desiccation. There are cells that you can desiccate down to nothing. You can modify human cells, and some insect cells on their own. You can modify, you can dry them. It's kind of fun working this field. When someone sends you cells, normally they have to pack it in dry ice, they send it to you, you pick it up at FedEx, you put it in the freezer. These cells you send in an envelope. You just pop it in the envelope and send it to the mail. You keep these cells in your drawer because they're dry. Nothing's going to happen to them. You add water and they grow. So I think we're trying to fashion tools that may be useful for long-term travel in space. Other people are trying to come up with ways in which lifespan can be extended tremendously. Will these be new species?
Potentially.
Potentially. But I think our mind is so creative. I think so far, as you said, the extremes that we see within our own solar system, if we can conquer those, it's possible we can conquer many other environments.
There's a famous mathematical evolutionary biologist named J.B.S. Haldane, who's English, who was once famously said, not only is the universe stranger than we imagine, it's stranger than we can imagine. And that's often been seen to be some sort of a limit on our cognitive abilities. Can we really? But the interesting thing is he said that, I think, in 1927 or 29. And since then, we've actually imagined some pretty strange crap that you never would have imagined we could have imagined. So that prediction has not actually come true. And I find that very positive.
Well, and I also think that people thought, when you're on those long missions, people thought you would just be at the whim of the universe and have to just be subjugated to this damage. But with CRISPR and genome repair systems, you could, much like you're continually repairing your ship, be constantly repairing your DNA on the way. And you would actually, I think we'd have to do that, actually. If it's a long enough mission, you'd have to have multiple generations that live and die on the same vessel before they get to the planet, which sounds awful. But if you have all of Netflix for the whole mission, I don't know. Might not do it.
And not a few things that you have to deal with here, too. Let's think of that, too.
Good speed.
We have time for another couple of questions, yeah?
What is your confidence that Mars will be colonized in your lifetimes or in this boy's lifetime?
By colonized, you
would
mean people having children and spending their whole lifetime there? Well, I'm pretty old.
I think it probably won't be in my lifetime. I'll be really happy just if we get there, and if my life's work has in any way contributed to help that make possible. I think in his lifetime it's possible. That would be the very first real colonist there, but it's a rough place as we've heard.
Yeah, I think it's awfully difficult to predict. You've got to but you've got to in this case you've got to predict the nature of human will, and the reality of chances are those first colonists, they're never coming back. I mean, that's certainly the Elon Musk plan, and it makes a great deal of sense because it's awfully difficult to bring people back again. But I think it's conceivable. let's say Tesla does really well in the next year, that within the next 10 to 20 years, there'll be an effort at least. There'll be some sort of attempt at an outpost. I don't know if it would be considered a colony.
We're out of time, but let me thank the panel, Chris, Ting, and Caleb. Would you please join me in thanking them for a great conversation? Thank you very much.